Okay, welcome back. So it's my pleasure to introduce our next speaker, Hana Aliee, who, I've already forgotten how to say the first name, who did a PhD in Nuremberg in Germany, and then did a postdoc in Fabian Thies's lab in Munich. And has been at the Sanger Institute in Hingston near Cambridge for about a year now.
Yeah.
Yeah. And she's going to talk to us about causally-inspired generative modeling for disease study. So over to you, Hannah.
Yeah, thank you very much for the introduction and for the opportunity. My background is in computer science, but then I was very interested in the application of my research in health. In this talk, I assume that you have heard a lot about causality in the previous talks in this school, so I assume that you're already familiar with the topic, and I want to show you how we can use these models for real applications, particularly in genomics and health, and why we need to actually change these models so we can't directly apply the methods simply because many of the assumptions do not really hold. So in this talk, I will talk about causality-inspired generative modeling. Maybe at the end of the talk, you can guess why I call it causality-inspired and not just causal generative models for studying diseases. So before starting, I would like to ask how many of you are familiar with single-cell technologies? Okay, quite many. That's good. So I have some introduction slides. So feel free to ask any questions. So in our human body, we have trillions of cells, and these cells are very different from each other sometimes, and they have different functions. So they shape together 78 organs, and so far we were able to classify them into 400 different cell types, major cell types. And still there are some cell types that we might not know, and they are being discovered. But if we look into a population, then these variations becomes even more significant. So our genomes are different from each other, so our cells varies. We might have some diseases or going through some treatments that all these can change our cells. So the main idea here is what is the differences across these cells and what makes cells different from each other. So within one human body, we only have one genome, one DNA in all the cells, but cells are different. So what makes them different? And that's the regulatory circuit underlying the cells that specifies which genes should be expressed in the cell. And these genes define the function of the cell. And these cells are different, and then together they can shape tissues, and then tissues can shape organs. And one of the questions in biology, important questions, is how these organs are developed and what are the regulatory circuits underlying this formation process. So with single-cell technologies, we take sample from a tissue, and then we are able to sequence the genes, for example, the transcriptomes, within each cell individually. And these transcriptomes can be used as a metric or measurement for similarities or differences across cells. So what we get as the output of these sequencing technologies that is interesting for computational people is a cell-by-gene matrix where cells are your samples and genes are your features or variables in the data. And the values in these metrics are the gene expression, for example, the number of times that the gene has been expressed in the cell. And from these metrics, we would like to learn about potential patterns across cells. And this can be done with some downstream analysis, for example, representation learning, and then unsupervised clustering of cells. We learn about these patterns, and then we can also find out what are the main features or genes that are expressed consistently across these cells, what makes these cells similar together, and sometimes we can assign these genes to some markers that we know in advance, for example, what are the markers for neurons, and then we can annotate these cells. So we know, okay, this cell is neuron, and this cell is, I don't know, some immune cells or something else. The Human Cell Atlas Consortium is a very big consortium that tries to collect all these single-cell data. And so far, there have been like 50 million cells that have been sequenced around the world. And these cells, 50 million cells, come from 7.7 thousand donors or individuals. So this is a huge amount of data. And the differences across these cells comes from a lot of different covariates. For example, the age and the sex of these donors vary. So we have samples from different ages. These samples might come from different organs or tissue. They might have, the individuals might have different diseases or going through different treatments. And also sometimes if we have developmental samples, for example, during the embryo, then we have other also covariates like time or space that makes these samples different from each other. These four main categories are the biological covariates that are very interesting for us to know how they make cells different from each other. Mostly because of the therapeutics purposes, how people are different from each other and so on. But apart from these biological variations that are very interesting to discover, we have another source of variation which is coming from the platform or the laboratory where the data has been generated. Simply the hands that have dissected the tissue or the hands that have sequenced the sample can already make these results different from each other. And sometimes we are having just different technologies, for example, different sequencing technologies that different labs use, or maybe the temperature, the room temperature changes from one lab to another, and all these can change cells because they are live sometimes. So this variation causes a lot of technical noise to the data, which is coupled with the other biological variations that we are interested in. And this makes the analysis of this type of data extremely challenging. So it's one of the main challenges in single-cell data analysis to decouple this noise from the biological variations that are interesting for us. So with machine learning, we are hoping to learn these biological variations and decouple noise. And we are hoping that this can help us to learn about the similarities and differences across individuals. That is the basis of personalized medicine. And after learning about these differences, then we can train these models to also predict, using, for example, generative models, to predict the effect of some, I don't know, treatments on some patients. So, for example, what would happen if we give this treatment to this patient? How would the cells respond to that treatment? Okay, so as I said, cells are different. They have different shapes, different sizes, different functions. And the first goal of my project is to dissect the source of variation in the cells. So first we need to learn a representation of cells that each point in this representation, two-dimensional representation, is one single cell. And then you can see some clusters of cells that are colored differently. And these are, let's say, cells that are very similar to each other and might associate to a cell type, a specific cell type, like, for example, neurons. But as I said, these cells have different, they're different from each other. Also, we can see that they, on these, for example, UMAP, they are located in different places. And these variations, sometimes it's just because of the stochasticity in the biological process. So two neurons in my brain, even though they have the same genome and they have the same function, and even if they are very close to each other, still they are different, slightly different from each other. And this is just a natural, stochastic variation across similar cells. But sometimes my neurons might be different than another person's neuron just because they have been going through some injuries, brain injuries. So disease can also cause cells different. So maybe some of these cell types are different from each other, some of these cells, because this is healthy and this is disease. And as I said, sometimes there is these differences between them just because there is a technical noise that we couldn't correct for properly. So the first question is, how can we remove these sorts of uninteresting sorts of variations from the cells? So the second goal of my project is making biology predictable. So if we can learn how the regulatory circuits underlying the cells work, then we can model this circuit with some causal models, for example, structural causal models, where the variable xi, which is, for example, gene I, we can write it as a function of other genes, the causal parents of the gene, and later we can intervene these causal parents and then predict the effect of that intervention on the cell response. So this will shift the distribution of the cell, and we can have a new cell type, and we are interested to know how cells respond actually to these interventions. And for this, we can use generative modeling. We can look into causality, and you can guess how this might be important. And also, I will show you an example where dynamical modeling can also help us. But there is another question that is even more interesting, is that what if we want cells to respond in a certain way? So how can we intervene these cells so that we get a response that we are interested in? So if we have cancerous cells, how can we treat these cells? So answering to these questions has a lot of therapeutic opportunities and also applications in drug discovery, as discussed in the previous talk. But why now, and where are we in this field? So these single-cell technologies were developed in 2009, and since then there have been a tremendous advancement in both the technologies. For example, initially it was just sequencing transcriptomics, but now we have DNA, proteomics, and other modalities as well. And also we have observed a significant improvement in the scalability of these technologies. For example, the number of cells that are routinely experienced in one single experiment. So up to now, we have a lot of samples, 50 millions, and in parallel to this, we are seeing advancements also in machine learning methods in different fields, for example, in language modeling or in imaging. But many of and these two are quite moving parallel and quite exponentially, both of them. But the question is that, can we directly apply these machine learning methods to biology? Previously, we have been doing this, and a lot of times machine learning could help to significantly improve. But now that we have more and more samples and more complicated data sets, because we have a lot of different sorts of variation, it's the time to really think of developing machine learning methods that really fit and are customized for this particular problem. Okay, so questions so far? Okay. Yeah, sure.
So many things, so I couldn't understand how do you represent these cells as? So is it always a matrix, or is it cells and genes together as one matrix, or how is this represented for machine learning purposes?
So for the machine learning, the input to the data, let's say I'm only focusing on transcriptomics, which I will only focus in this talk, then it would just be a cell by gene matrix. Cells are samples and genes are the features.
And when you showed the plot of the two-dimensional plot of cells
So this is just a representation of the data. So, so initially we have, I mean, in human bodies we have 20 to 30,000 genes, and we need some dimensionality reduction methods to reduce this to 20 or 30 dimensions. And then from these 20, 30 dimensions, we use some visualization methods, for example, here UMAP, to represent the data in 2 dimensions.
Okay, thank you.
Yeah, sure.
And also, how do you define these parent genes mentioned in the studies?
This is what I will be discussing next. Yeah. Okay. So to reach to my goals and the questions that I discussed earlier, the first step is to understand what are the cellular changes. What happens if cells goes through some sort of intervention or perturbation, we call in biology. So there are 3 main cellular changes that I will discuss here. The first one is that sometimes we observe that after a perturbation, such as a disease, the composition of cells changes. So, for example, here the number of red cells reduce, and then the blue cells are overtaking. And this actually happens in some diseases. For example, with leukemia or blood cancer, the cells from bone marrow, they lead to the blood, and then they change the compositions of cells in the blood. So by observing the compositions of cells, we can diagnose the patient with a disease. So it's already a way of diagnosing, or we can observe, for example, how the patients over time are responding to treatment, because we expect certain cell types to reduce with, for example, chemotherapy in the patients. So I have some methods developed for this purpose, but I'm going to skip it for the sake of this presentation. But the second category of cellular changes happen when a perturbation like disease changes the regulations of cells. And this is a quite common case with many diseases, that some certain cell types might change. And we are interested to know what genes were targeted by the disease and how these regulations changed. And last but not least, sometimes during development or regeneration, we also observe a gradual changes in the cells or in the regulations of cells. And we want to know how, for example, an embryo is developed and how we as a mature individual have been developed from one single cell. And here we have time and space also as 2 dimensions. Okay, in the rest of my talk, I would like to first start with answering how gene regulation changed during development and regeneration. So, as I said, we are all generated from one single cell, and this single cell divides and differentiates into more mature cells over time, over the developmental time. And one very fundamental question in developmental biology is that how cells decide their future. So how do they decide what to become? And more importantly, what are the genes that are leading this process? Because in our human body, we have like 20,000 genes, but we only have something around 400 genes or transcription factors that are leading the other genes. So it's important for us to know what are these leading genes for each of these developmental processes. And at the end, at some points, we will have mature cells. And also, if we know how this works in healthy, then we can also look into the diseases and see how cells diverge from diseases, how the regulations change that cells diverge. And then for designing new therapies, these genes help us to develop new drugs, so the target of the drugs. Yeah. So what we can get from this type of data is the gene expressions over time. And one of the causal questions that we can ask is if and how gene 1 caused gene 2 in this temporal process. And if we intervene this gene, for example, by knocking it out or turning it off, what would happen to gene 2? If there is a causal relation, we would expect them to change together. Okay, but this is in theory, and it sounds very nice, but in reality, when we use single-cell temporal data, things are more complicated. So first of all, with single-cell data, we have a snapshot. So we take a tissue, an embryonic tissue, and this tissue has cells that, from different developmental stage. But with single-cell technology, we mask all this information, so the information about time and space. So we have to recover this information from the data. We need some manifold learning methods that can learn about based on some similarities between the cells, they can build for us how the cells might have been developed. And then if we are interested in a certain part of this data, then we can take it and sort the cells along that trajectory, assign a pseudo order or pseudo time to them, and then we can, from this, we can then say how gene, for example, X and Y here, their expression changes over pseudo time, and from that we can learn the potential interaction between genes. So the point is that we don't have real time. We need to infer the pseudo order or pseudo time from the data. But what's the complexity? Okay, let's assume that we can learn these pseudo orders, but still, this is quite complicated. So in one of the projects that I was doing with my collaborators in Boston, we were interested to know how the pharynx organ is shaped. And this is an organ that is very important for the development of the thymus tissue, which is related to immune system. And many kids born with some immune disorders that is associated with the development of the thymus tissue. So we were interested to know how this thymus tissue is developed and what are the target genes during, what are the transcription factors affecting that development. So the data that was given to me was something like this, only for a very tiny small part of the data, which was for pharynx trajectory. And I'm showing you here only very few genes, like 20 genes. So you can imagine how this would look like if we had 20,000 genes. So this data is quite high dimensional. And already you can see the order of cells along the trajectory. The data is also noisy, so it's not super clean. But by just looking into that, we can see that some genes are upregulated in the beginning of the trajectory, and then they go down, and some genes are low expressed, and then over time they go up. So the question that I was asked was, can you tell us what genes are causing this process? It's a super complicated problem. It's it's a $1 million question in biology. And I couldn't really answer it properly, and because most of the methods in the literature at the time, they were looking into the correlation between genes. So are the genes co-expressed together or not? So what they build at the end is a correlation, kind of a graph. They call it gene regulatory network. But in reality, there is no causal relation between them, so these are not directional. And this is because it simply is based on correlation or some sort of mutual information between genes. So this could not help us, of course, because these methods often ends in a lot of false discovery edges in this network. But but what we were interested in was some things that look like this. So we have transcription factor only causing target genes, and also sometimes the edges are directed, and also sometimes we have self-regulations because some transcription factors can regulate themselves. And with the previous works, it was not possible to learn about these cyclic dependencies. So I was like, okay, this sounds like a perfect problem for causality, so let's look into these structural causal models. With most of the methods in this domain, they focus on static structural causal models, where we model the gene expression as a function of some causal parents. So if we can learn these functions with some DAG constraints, the directed acyclic graph constraint, then hopefully these variables that enter the function are the causal parent of our target gene. Okay, it looks very nice. Then the function f is the causal model, and the variables inside the function are the causal parent. So from this, what we get is a directed graph in a very ideal case, but still in static cases, we don't get any information about the cyclic dependencies between genes. So to recover those cyclic dependencies, I look into the dynamic structural causal models, where we model the time derivative of the genes or their velocity as a function of other genes and time. So with these dynamic structural causal models, we are actually able to also learn the cyclic dependencies between genes. Okay, everything looks great. The first assumption here is that we should assume that the gene expressions jointly evolve according to an ordinary differential equation, which is not a very bad assumption. A lot of previous work had already assumed that the gene expressions can be assumed to follow these. So we stick to that. And yeah, and then there was this also a paper from 2013 from Bernard Jacob's lab that they discuss the fact that the function that this function f, the ODE graph that we can infer from the function f, is actually the true causal graph. So everything is nice up to here. So we were like, okay, how can we model f? And then there is a neural ODEs that is a breakthrough method that tries to learn the function f from some time series data. So the idea is that there is a neural network that models the function f, which is the velocity, and then with some ODE solvers and the initialization, we can learn the predicted values or reconstruct the values and use mean square errors to train this function f. So it seems like a perfect match, and it has already been shown in the literature that these models have a very good performance, predictive performance, when we have irregularly samples or noisy data, for example, as you can see on this spiral example. However, we show in our preprint that these models are not identifiable. It means that they do not necessarily learn the true function that we use for generating the data. And one reason for this is that we are only giving a snapshot of data to the model, and there might be a lot of different functions that can properly predict the samples, and they might just, for the model, there is no preference which one to select. So yeah, given the observations, we could not identify the function f, and this is then this is where these models break when we want to use them for our purpose, which is for learning the causal relations. Because if they are not learning the true function, then what are we learning? This has no meaning for us. So, and the other problem is that because we are using neural networks, they are they are known to to converge to the fully connected networks, and therefore this is also another problem, that at the end, the system that we learn is a causal system that everything is connected to everything. So one solution to this ill-posedness problem is using regularizations. So the regularization helped to reduce the search space, and maybe with some prior knowledge, we can hope that the model converges to a solution that we are interested in. So the question was, what is the right regularization? In causality, sometimes people assume that the causal model should be sparse. It means that the number of variables that enter that affect the system should be few. And this is simply because nature prefers simplicity. At least we assume this. And in biology, it's also quite true, because we have fewer transcription factors compared to the target genes, and also within each tissue, the number of transcription factors are significantly less than the target gene. So it seems like this sparsity regularization is a quite a good assumption in biology. So I stick to that, and then there are different, there are millions of methods on sparsity, but I could not directly use them here. So the first group of sparsity methods focus on weight sparsity. They try to sparsify the weights, individual weights in the neural network. But if the function f, which is represented by the network, is going to be sparse regarding the interaction, still you can see that with weight sparsity there is no guarantee that we are sparsifying the input to output interactions, because still, for example, this input can find a way to affect the output. So these methods do not necessarily make this cause these outputs to be sparse. So we have also feature sparsity, and these methods try to sparsify the features, but these often only predict one output at a time, and this is why they work. So they work very well, but then if we have a lot of different outputs, for example, if we have thousands of genes that we want to predict, then we need to replicate the network 1,000 times, which is not very efficient in our case. So I was thinking if we can combine these to have only one single network, but ensure that we can reach feature sparsity. And for this, I propose pass sparsity, which sparsifies one complete pass from input to the output. And this is a more aggressive sparsity compared to the weight sparsity. And I show later also with the results empirically that this can increase feature sparsity significantly. So what we hope to get from this, because this is the network that is used in a dynamic model, then hopefully is the true causal graph that we expect. And to train this model, we develop some. So we use some stochastic gates, z, as a function of some random variables s with a distribution q, and this z is just the mean of 1 and x, 0 and s, which means that if s is negative, this z gets 0. The output of the gate is 0. And this was very important in this application because often when we use regularizations, such as L1 or L2, the aim is to reduce the weights, but they never get to 0, because this is the problem with the optimization of the neural networks. They try to converge to 0, but they never get 0. And if we want to really know if this gene is affecting the other gene or not, we need sometimes to make the cutoff, like, okay, if the weights are below this, then we assume them to be 0. But this was not very nice, and we really wanted to achieve 0. And with this definition of stochastic gates, and with this function z, which is differentiable, we could get exact 0. So it's an implementation of L0 regularization that's differentiable, in summary. And then we can calculate the probability that the gate is not 0, and then from this we can calculate the probability that the whole pass from input to output is 0, which is the multiplication of the probabilities of gates through the pass, and then the constraint is minimize the number of passes that are active from input to output. And applying this to some synthetic data, here I'm showing you, this is the second order differential expression, so we try to predict the acceleration as a function of position and velocity. And if this is the base model where we use no regularization with neural ODEs and passreg is my method, then the first thing that we observe is that in the adjacency metric, which represents the connection between input and output, our method, maybe it's too small, sorry if you can't read it, but we achieve exact zeros, which is very valuable in this problem. And also looking into the reconstruction, except L0 that failed, but most of the methods work very well in building the reconstruction, even though the function that they learn was actually wrong. But when we look into the extrapolation accuracy, then we could see that only passreg that predicted the adjacency metrics properly could extrapolate also with a high accuracy. And I applied this also to motion capture data, and simply because, not because this was submitted to Norris, which was one reason, but also because with biological data it's very difficult to validate this interaction because there are a lot of unknown things, and this application could help us to visually validate the results. So the idea is that when someone is walking, the hands might move with the legs, and we try different motions, and we wanted to know if the model learns the true causal relations between variables. So hands move with the legs, but if I hold a box, then my hands is not maybe consistent with my legs anymore. So there should be no causal relation between my hands and legs. But this is how the extrapolation looks like for the passreg method and for the base method. And you can see that for the trained data, already the predictions are not that bad. Reconstruction is not bad, but extrapolation starts getting very off for the base model. And if I continue this, then at some point the person explodes. And then with the test data, this was also similar. The predictions were even worse for the base method. But then looking into the adjacency metrics, it was very interesting for us that we could learn that the legs do depend on the legs, but they do not really depend on the hands. And this was very interesting for us to see. But if you look into the adjacency metric from other methods, you can, for example, see here that there are a lot of non-zero interactions, and this is why they are not very good in extrapolation. Okay, now let's go back to the biology and see how these methods can help us to identify the relations between genes. So here I looked into the mouse gastrulation single cell data, and I only look into a branch of data which, in which blood progenitors differentiate into erythroid cells. And you can also see the pseudotime that I assigned to this data. And in this particular trajectory of cells, it was known in the literature that there are two transcription factors, GATA1 and GATA2, and I'm showing you here their expression over the pseudotime, that they affect the expression of hemoglobin genes that I'm showing you in this second plot. And if we just visually look at these plots, we can see that these genes here are more correlated to each other than to the transcription factors. But when we apply the method, we could see that the velocity or the rate of the hemoglobin genes depend on GATA1 and GATA2, and they do not depend on themselves, which was surprisingly very accurate. So we were very happy to see and validate these results also on single-cell data. But this is a very small data set, so I have to mention this. And this was just because if you increase the size of the data, there is no ground truth to really validate, and that's one of the main problems with this problem, actually. So, but still, we wanted to see how it works if we extend the data. So I looked into the human hematopoiesis single cell data, where the stem cells differentiate into 3 different lineages, as you can see here. And I'm showing you only 3 genes per branch, and their expression and also the prediction of the model. So it seems like the prediction works well, even though the data is quite noisy. But this is not enough for us. There is no causal evidence in these results so far. And looking into the sparsity, we can also see that pass rate could both increase the weight sparsity and also the pass sparsity. So it's nice. We are getting a sparse interaction between genes. But what does this mean? Here I'm using 2,000 genes, and this whole network is super massive to look into and digest. So one feature or one main characteristics of causal model is that if the model really learns the true causal relations, it's more generalizable than any other method. So to test this, I trained the model only with one single hematopoiesis data set, and I tested it on 4 other samples. And these samples were coming from different patients, and they were sequenced in different labs. So there were batch effects also between them. And I showed that pass rate is the one that gained the highest performance and also the highest sparsity. But still, this is not biological validation per se. And I defined another sparsity feature, which I call it spurious feature sparsity. We know that only transcription factor should cause target genes. So there should not be any interaction between target gene and transcription factor. So I tried to find this, count the number of false positive edges that these methods could find, which is from the target gene to the transcription factor. And then I show that my method could maximize the number of sparsity between target genes and transcription factor, which is another validation for the method. So, questions? Yeah?
Maybe it wasn't clear to me, but when you were defining the function, what were the constraints that the function has to have, like 0, so it's sparse, which was clear, but did it also have to be Lipschitz continuous?
Yes.
Okay. How did you train that thing at the same time being sparse and Lipschitz continuous?
Sparsity is added as a...
Yeah, I get it, but does the gradient descent work in the sort of setting? Or do you have a different training way for this?
No, we are using also stochastic gradient descent for training. Yeah. Yeah.
I don't really understand in what, I guess, circumstances with regard to like the data generating process, you would expect your model to be a causal model? Because I thought there had to be some like magic fairy dust about like assuming certain things. Could you clarify?
I'm not sure if I got the question. So.
So I guess like I don't. As I understand it, you're like fitting a neural ODE. And I'm wondering, in what scenarios is that guaranteed to be like the true causal model, right? As opposed to, I mean, how is it better than just doing, let's say, some other form of regression?
Oh well, yeah.
Causal aspects, yeah.
Yes, well, there are different reasons why I use neural ODEs. First is to, first of all, it was about this. About this dynamic feature, which can let us consider cyclic dependencies. So this was why we went from static to dynamic in this work. And then why neural ODEs? It was just an option for me to model function F.
But are you convinced that you're inferring the correct direction of causality, for example?
Yeah, this is what we have been trying to prove empirically. I mean, also with synthetic data, we could show that it, I mean, this, for example, these values here. So we define some ODEs. So the coefficients in the ODEs we are trying to predict with the model. And this is exactly what the coefficients are summarized in these metrics. So for a series of synthetic data, we show that the model can predict either accurately or better than many other methods. The coefficients from the ODE directly.
But you don't have like a general statement about like, okay, if my data has such and such structure, then we feel that this is going to yield the correct causal model, excluding other kinds of structure.
That's a very good question, and we have been working on this because even though we are adding regularization, but still the problem is ill-posed because we are adding the constraint to the loss, and they together should be identifiable, which I mean, we can't guarantee that. And we have been thinking of finding the functions that can be modeled, or at least where this sparsity assumption, where does it really hold? And if we add this regularization, is it for what type of function is going to converge to the true system at the end? Yeah, but this is still open for us. It's not very easy to answer that.
So I thought with these structural causal models, like we had a talk about them earlier in the summer school, that as long as the function is nonlinear and the noise is independent of the function, and you have sufficient data, you can kind of guarantee some kind of identification of these types of causal models, like some conditions on nonlinearity of the function. But I don't know if that's true. I mean, obviously, if you have noise, and you can't necessarily identify things, so you might need a huge amount of data for that to be identifiable. But is there some conditions about general nonlinearity plus Gaussian noise sufficient?
That's a very good question. At the time, I couldn't really find a reference that could, we could use to really prove things, because the other question is, what is the noise model in this single cell data?
Yeah, it's not Gaussian.
So exactly, it's not Gaussian, and it's also, if you have batch effect, then things might change. But I love to continue this research, but unfortunately at the time we couldn't really summarize. But if you have ideas, I would love to hear more.
On your 18th slide, this slide before this. Okay. Yeah, this one. In the first equation, you have given a noise term, but in the neural ODE, there is no noise term, right? So is it an SDE or?
There is no noise, yes, in this equation that I wrote. That's right, but with this sparsity, we are kind of adding noise with the sparsity method, because the sparsity method is probabilistic, and it learns the probability that the passes should be 0 or 1, because the gates are probabilistic. And we have to sample from these models. So it makes, it's add.
Would this become an ODE or an SDE?
If you set the weights, then it would be ODE.
Okay.
It's not, Yes, it might look like STs if you select the probabilities, but no, I mean, no, by definition it might not be STs. It's just ODE.
Okay, that was one question. And another one is, you have a set of initial conditions, and then your x of t is this final flow, right? The last one. So when to understand what you mean by the path sparsity, do you mean some of these initial conditions are not required, or some of the right-hand side coefficients are not required? What is that?
Is this clear what I mean here with the sparsity, with the few number, and how this is related to the neural network? Is that, so these dependencies between variables, this is what we infer from the network. So if there is, if through this network, if all the weights are non-zero, then it means that all the outputs depend on all the inputs.
So the inputs are initial conditions?
Inputs are genes, all the genes, gene space, and output is also the gene space.
Okay.
So I try to predict genes from genes.
I think I got this. I think I can take it offline.
Yes. Yeah.
Could you go to your loss function? How do you define here, for example, the hyperparameter S? Like, how do you infer it from the data?
The hyperparameters for the gates,
Yeah.
they're just like trainable variables in the neural network.
Because on the next slide, you're comparing, first of all, like you're working for two things like nonlinear versus linear methods. And then, for example, in the last one, what was the hyperparameter for the regularization? Because you have the reconstruction loss and the regularization loss, and you can scale the regularization loss to like with different hyperparameters, and then you can enforce zeros.
Yes.
Do you also learn this parameter?
For Lassonette.
Yeah.
Oh.
Because if you have a super high regularization, then you go to zero, given the reconstruction loss.
For all these methods, if I'm I'm not sure if yeah, I have to recap because I did this like 2 years ago and I don't remember the details of the Lassonette, to be honest, now. I have to think about it. But what we show here is just the weights that we learn from the network.
Like, I'm not sure, is the weight like a sum of the weights, or
It's all the weights that are from the inputs to the outputs. So we are predicting the acceleration using velocity and the position, and this is all the weights that we get from the network. So we are only focusing on the ODE that these methods have inferred. Sorry.
yeah, because normally in the last net you have the hyperparameter with which you can exactly control how big your weights will go eventually. And if you have a high parameter there, then you're enforcing the zeros. This is also the last net.
I would like to chat with you later because as long as I know they don't converge to exact zero, but maybe there's a newer version.
Yeah, I just wonder, like in your model, you have the explicit dependency on the time for parent and time. So I was thinking that maybe the time variable would be also incorporated into the a-causal variable of your model, so that you don't need to explicitly mention it because your parents of the variable would also include the time variable as a positive factor. And And then also wonder if you have some time variable in your model as input to your neural OD to correctly estimate the behaviors to model the time dependency.
In these methods, we use autonomous ODEs. So time could help us sometimes with the biological problems because if we had, for these problems, because we had different branches, and here the time scales varies between the branches because some, for example, one branch of cells might have faster development compared to the other ones. And here we use times as, we can use time as kind of a condition for the model to also specify different branches because this is ODE-based method, it's not STE, but we are conditioning the model on each branch. So yeah, it can potentially be used.
Yeah, but for now you didn't use
No.
time. So my question was, like, in that case, like a time is also could be a kind of causal factor you identify.
Yeah.
Yeah, maybe you can simplify more into more complex form.
Yeah, potentially, yeah. But it can just complicate things, and in biology, we don't really know what is that causal relation. So we try to keep the methods as simple as possible. Yeah.
So you now validate your gene regulatory networks based on the relationship between a transcription factor and a gene, but you may also find relationship between genes or between transcription factors. Is there any way that you can validate those relationships?
In larger scale, no. Just simply because there is no ground truth. There are more unknowns than knowns, so yeah.
And on small scale you can?
In a small scale, yeah. For example, with this one, yeah, because it was already known. So they do change these genes in the lab, and then they see the effect. So this is why we knew how this system works.
When you're, I have another question on the hyperparameters. When you're setting the hyperparameters of your model, do you need to look at the sparsity you get out at the end to evaluate whether the model is good or not, or is just the test accuracy kind of sufficient to judge whether you've got a good set of hyperparameters on your model?
For this analysis, I looked into both actually. So the reconstruction error was important, but also sparsity, because we knew that the graph has to be sparse as well. Yeah, but it's not multi-objective. If this is your question, it's at the end it's the sum, but I always check if it really causes a sparsity. Okay. Yeah.
I'm not sure what slide number that was, but somewhat results-ish, not this one. There's somewhat a notion of performance or overall performance on that slide. I've been wondering, is that reconstruction or somewhat an aggregation of different?
Here, any of these?
This one?
This one?
Yes. The one before, I think. Yes, the overall performance.
No, this is overall performance over train and test data. Yeah, but I mean the performance for each test case is actually shown here, and this is the average.
Oh, nice.
Yeah. Okay. I move forward. So to the last part of my talk, which is on how these perturbations can change the regulations in the cells, and if we can, how can we actually learn about the cell states that are actually shaped with these perturbations. So this is a quite quite of a challenge in single cell that when, for example, the disease has affected the cells, sometimes the differences are so subtle that many of the representation techniques can fail to learn these differences between disease and healthy. And so I would like to know how we can improve this. And by cell state, I mean, so we have cell types and we have cell states. There is no very clear definition between them. But by cell state here, what I mean is that a very subtle differences within the cells of a certain cell type. So let's see what's the challenge of representation learning from single cell data. So if we have a sample, a control sample, for example, a healthy sample from donor 1 and batch 1, and by batch I mean the experiment. Then we also have another data set which is the cells, similar tissue but with some sort of perturbations. And we would like to integrate these two samples together and learn a joint representation that should capture all the biological signals in the data. So looking into this example, we should, the good representation learning method should know that the blue cells are very similar to each other in both of these experiments and should group them together. But it should also learn the disease variations, which is in cell type 2, we have some red cells that are healthy and some cells that look different than this one. So it should be able to learn these disease variations, which we can call it also invariant to noise. This is also one way of formulating it. It's invariant because for this blue cell, it has decided to, it has learned that these cells are very similar, even though there is technical noise between them. And this method should be interpretable. So we can learn about the cell types, and also it's identifiable, meaning that here is slightly different than the identifiability I discussed before. Here I mean by identifiability the fact that if we retrain the model with a bit of a different initialization, we can ensure that the model can reach to a more or less similar representation. So this is what I mean here. And then this was a very ideal situation. But what happens in reality with many of the methods is, first, many of them can undercorrect. So if you just give all the data to a variational autoencoder, then what happens is that often they learn two very big clusters of cells that they are separated mostly because of the batch effect, because batch effect is quite significant, and batch effect is the technical noise between the experiments. It's so significant that it can easily dominate the clusters of the cells. So this case, they are undercorrected, and another very common scenario is that these methods might remove the noise, but they can overcorrect for noise. So the similar cells are grouped together, but also the disease and the healthy cells are also grouped together, because the method could not really separate and learn the disease variation between them. So this representation learning is therefore is a challenging problem, but what we hope, if we can find the ideal method, what we are hoping to achieve is a reference atlas of cells. And this reference atlas can be built using, for example, healthy data that we can later annotate the cells, and then whenever we have a new data set, we can map this data on top of this reference atlas, and then annotate these new cells. So they often look for similarities and for mapping. But one issue with them is that if the differences are very subtle, again, these methods might fail to map the different cells. For example, here these light blue ones will be mapped on the red ones, but this is actually a new cell state or new cell type. So this is, again, why we need a very good representation learning method that can consider that this new data set, for example, is coming from a new condition, is a new disease or something. So you should have this in mind, that if there is a new condition, then there might be some new cell states. And for modeling this type of problems, most of the methods in the literature are statistical. And here, for example, if we have cell types and different perturbations, like, for example, adding different drugs to these cell types, and we train this as a statistical model, of course, they can be very good in reconstructing the data, and also for predicting the performance of the data that similar data to what they have been trained on. But of course, if the environment is very different, for example, we have a drug that was not seen before, this statistical method failed. So this was, again, one of my motivations to look into causal models and try to see if these causal models can help us to have a more generative or predictive models. And many of these challenges we have been discussing in our very latest review paper, which was a collaboration with Yoshua Bengio from Mila. So to train this type of causal models, of course, we need two different types of interventional data. First of all, we need to have enough different perturbations. For example, these perturbations are biological variations. And also we need different replicas. And replicas is important because they are modeling the noise or the batch effect in the data. So from each experiment, when we have multiple replicas, the model can learn the noise model to separate it. So this is the type of data that we often need for training these causal models. But at the end, the generalization is always limited. So it means that because we can't really have all the possible perturbations, it's massive and it can be super expensive. So at the end, what we gain is that if we train the model with only with these few samples, then we will have a generalization property that is still limited, meaning that the model cannot predict everything, whatever that is possible. But hopefully, the more data we provide to the model, the more we can expand the world for the model. So this is also something that we should have in mind, that we might not be able to capture full generalization anyway. And one of the other challenges with these interventional data in real case is that if we learn a causal graph from the data, and we want to intervene a gene and see if the intervention, what are the causes, what are the genes that are affected by intervention. What we ideally assume is that this intervention is perfect. It means that it's gonna change all it's gonna only target the specific gene that we are interested in, which is G4 here. But in real cases, this is not really possible. So a drug can have multiple targets, and also sometimes this can be imperfect. And even in a worse scenario, sometimes they might have some effects that we even don't know or we haven't anticipated. So this is one of the challenges with real interventional data. So with all this, the main question is that how can we, or can we even, learn a true causal model from a system? Do you have any idea? Yeah, so to answer this, I mean, we would like to learn all the interaction between genes, but the complexity is that this interaction changes from one individual to another. So if you have thousands of samples, how should my gene regulations differ from another person? And also, if the interventional data is not perfect, how am I going to consider all this with technical noise and so on? So it's like, boom, it's super, super complicated to learn such a fine-grained causal graph from the data. So in the rest of my talk, I would focus on methods that we can, that's causality-inspired, and this is why I call it like this, because it tries to learn a model that is close to a causal model, but might not be the true one, for clear reasons. So before that, let's see what happens and dissect the problem a bit more. So in a pathology, if we have a control and case, and let's say the case cell has been affected by some condition, a virus, and if so I observe a shift in the distribution of cell, clearly, but if I know that this case was experienced in a different lab compared to my control, so now I have two events that are affecting my cells, the condition and the batch. So I don't know if the shift in the distribution is actually because of the batch or is because of the disease. So which of them has really caused the cell to respond in a certain way? And this is a question that we can't easily answer if we only have very limited data. But if we aggregate data from different environments, then we can say that the non-causal variables are affected by the environment, some spurious correlations, but the causal dependencies remain invariant. This is one assumption beyond invariant representation learning. So I can try to learn a representation of the data or a causal model that is invariant to the noise or all the spurious correlations. But how does this work? So to model this, I came up with this idea of a generative process that we have two sets of latent variables. The first set is invariant, and we assume that these latent variables are invariant to noise, and also they capture all the biological variation in the data. And these latent variables are affected by some biological covariates, for example, healthy or disease, age, sex, and all these things, all these variables. And I have also some spurious latent variables that these are affected by the environment or by the batch effect. And then at the end, my observations is an aggregation of these two latent variables. So I need them to predict my outcome. And then from the invariant representation, from the invariant latent variables, we can learn the invariant representation, which should hopefully capture the variations across cell types and also different conditions that we have specified here. For example, here I'm showing you disease, but it can be also age, or it can be sex or other type of variations of interest. And then using also the spurious latent variables, we can also have a spurious representation here that should capture the technical variation between the data. And some things that is important to consider is that there should be a partial independence between spurious and the invariant representations that we apply using some regularizations. And also, often we assume that the latent variables are orthogonal to each other, so we add some regularizations to make sure that they are independent from each other. But in many real applications, this is not the right assumption, because if, in biology, for example, if these latent variables should represent a function or a process, a biological process in the cell, then many of these biological processes are dependent to each other. So the assumption that the latent variables are independent doesn't hold in biology, at least by definition. So to make this actually more general, these latent variables in the invariant part, they follow the general exponential function just to let them be dependent also. And this is how the architecture looks like. So here we have an encoder is similar to variational autoencoders or identifiable VAEs if you are familiar with. So there is an encoder that gets both the gene expressions, the biological covariates, and the covariates that are explain the technical noise, which we model it using one hot encoder. And then there are we learn two latent sets of variables, Zi and Zs. And these two should be orthogonal, but one main difference with variational autoencoder is that here the prior is not set to mean 0 and 1. We try to infer this distribution of the prior from the data. And what we use here is the covariates are used with an encoder to predict the mean and the variance of the prior distributions. And this prior distribution is very helpful because at the end, for each of your samples, you learn a distribution. So that's also sometimes helpful. And then we have a decoder that uses both Zi and Zs to learn the parameter of a negative binomial distribution, which we assume the single cell data has been generated from, and then we can sample from this to reconstruct our input data. And with this model, I tried integrating both healthy and disease samples. And here I'm showing you the whole human lung cell atlas that has more than 1 million cells. And the first thing is that each color is a cell type. That was, it's like 65 different cell types in this data set. VAE could properly detect these cell types and cluster them together. But apart from this, it's one of the first methods, or at least when it was developed, it was the first that could let you integrate biological covariates and therefore both healthy and disease at the same time in one representation. And I looked into airway epithelial cells, and here I'm coloring the cells based on their disease. And it's not important what these colors are exactly, but there was one cluster of cells that popped out in the VAE data that was interesting for us to know what is this disease and why this is separated from the rest of the cells. And we could actually also see that with related works, for example, SCVI, which is a very famous method of the field, we could see that these cells were spread all over the manifold, meaning that the method could not learn to separate them. But with VAE, we were able to separate them. So we look into the differentially expressed genes between these cluster of cells and the rest, and we observe some cells that are associated with this chronic disease. So it kind of validated that this cluster is really different than the rest, and it was nice that in VAE could learn these very subtle differences between cells and separated them. But as I said, this was the invariant representation of the data. We can also look into the spurious representation, and this shows you, for example, the similarities between cells, which is based on the spurious correlation between them. And I have colored them based on the ethnicity, background, and smoking. So, for example, it can help you with designing your future experiments. Here, for example, I see that there is one class of African ethnicity that is very separated from the rest. But you can also see that it's the only one that has the history of marijuana. So the question is whether this separation is really based on ethnicity or is because of the smoking condition. And if you want to design your experiment in the future, you can, maybe you want to focus on this and say, okay, I want to have an African that is not smoking, so I can better learn what is the effect of these variants. So in another example, I applied this to lung cancer data. And we have 3 conditions here. Lung adenocarcinoma and small cell lung cancer, and also normal cells. What we expect is in this data is that in the epithelial cells only, there should be a separation between a small cell lung cancer and the rest. And also a small cell lung cancer is something that affects the patients differently. So the data should change from one patient to another. So this is the representation from our method. And what you can see is that for all other cell types, the samples are merged, which means that the method did not separate them properly. Not properly actually, did not separate them, which is right, because we don't expect differences in other cell type. But only looking into epithelial cells, we can see that the orange cells are very different from the other ones. And when we look into the donor, you can also see that the donor are merged for other cell types, but they are very separated in epithelial cells of the small cell lung cancer. And that's very nice results, because if we color these UMAPs based on some genes that we know as markers of small cell lung cancer, then we see that these markers are active, highlighted, if you can see, in different part of these UMAP and for different donors. So some of them are, for example, ASCL, for example, is active in these patients and is not active in another patient. And this explains why the model has separated them from each other, which makes a lot of sense. So with this, I would like to just conclude my talk. So here, as I explained, the main goal of my research is developing generative models of cells. And here I'm interested in modeling the cellular heterogeneity, learning the causative role of genes, and also trying to predict the phenotypic effects of perturbations. And this is a quite interdisciplinary work which combines machine learning, biology, and also data science or statistics. And I would like to also acknowledge my previous group, Fabian Theis in Munich, and particularly Ferdinand Kopp, who was my master's student, and together we developed this method. Yeah, I'm co-supervising him. And also all my other colleagues at Fabian Theis lab, and also in my current group, Rosa Rovento, and also in Yoshio Benjo's group, and all my other collaborators and donors and families. And yeah, thank you very much. I would like to take any questions from you.